In lecture one of the urinary system, we looked at the renin-angiotensin system and how renin activates angiotensin 1 and eventually causes vasoconstriction, increases thirst, reduces urination, and which helps with blood pressure. So tubular reabsorption, we've talked about filtration and what liquids leave the capillary system of the glomerulus into the Bowman's capsule. As this filtrate moves, we are going to move useful substance from those renal tubules back into the blood system, into the peritubular capillaries. Peri means around, tubular capillaries. These capillaries around the tubular system. And so while some things were filtrated out that were good, we want to, absorb that back into the blood. And so most of this happens in the proximal tubule, and it has microvilli. We've seen microvilli before. We know what microvilli is for. It's for increased surface area, right? And different parts of the tube reabsorb different substances, and those substances go by cellular transport. And so diffusion, which is the movement of molecules from high concentration to low concentration that are lipid soluble. They can pass through the cell membrane. And then another type of passive transport, which is called facilitated diffusion, where molecules that aren't lipid soluble have to go through channels. So these would be ions and even carrier molecules that transport more than one substance at a time. And then osmosis is passive transport of water, and that water is moving from where there's a hypotonic solution where there's a lot of water towards an area where there's a lot of solute. The solute attracts the water. And so, yeah, we're using passive transport, but we're also using active transport. And in active transport, you're not going from where there's more of something to where there's less. You're actually moving against the concentration gradient. It requires energy. And so we're going to need glucose and oxygen. That's why this is highly vascularized, because we've got to make a lot of ATP with our mitochondria to facilitate this active transport process. Now, active transport is limited because of the number of carrier proteins. So what's going to happen is ATP is going to bind to one of these pumps, these what they're called carrier proteins, and that energy is going to allow these pumps, these carrier proteins, to move specific material up their concentration gradient. The more proteins you have to do that, the more transport you can do. Each substance also has a transport maximum. And so what this says is you can only transport specific number of molecules in a certain amount of time. And my students sometimes have problems understanding this, but it's kind of like, I'm a runner, so I think about running. If you asked me to run for 5 feet, I wouldn't get very fast, right? Like, I'm limited. But if you give me 10 feet, I can get a little faster. But if you gave me 5,000 feet, I wouldn't continually be faster. There's a maximum that I can run, no matter how much space or mileage you give me, I'm going to go at my fastest pace. Well, that's what they're talking about here. You can only move so many molecules at your maximum, even though there might be way more molecules to move. You're just going as fast as you can. And so the renal plasma threshold is reached when there is more transported substances in the plasma than in the active transport mechanism that could handle. And so what they're saying here is you have all this material that we're wanting to reabsorb into the blood that's in this filtrate, if you will. And there's pumps, man, they're trying to get all this good stuff back in the blood. But as the fluid is flowing, we're not going to be able to get it all. And so some is going to be seen in the urine. And so what are some substances that are reabsorbed by active transport? Well, glucose, amino acids, lactic acids, citric acid, phosphate, sulfate, calcium, potassium, sodium ions. So glucose, you might think of diabetics have glucose in their urine. Amino acids are going to be from protein. Lactic acid is from anaerobic respiration, where you don't have oxygen and you're trying to metabolize food. Citric acid is a chemical found in your Krebs cycle, and then all of these are different ions in our foods, right? So here's a visual for you. Here we're at our afferent arteriole coming in, high blood pressure, and we release this mainly small molecules, right, and water. And so what's going to happen? Not all, yeah, there's waste here, there's maybe toxins, but there's also a lot of good stuff like those ions, like sugars, like amino acids, things that we worked hard to get, or we should have, because animals work hard to get their food. So what's going to happen is we're going to reabsorb the good stuff. And remember, not everything that we needed to get rid of got out of the blood. So that's why we're going to secrete from the blood into the tubule, and we called that secretion. So we're going to secrete and reabsorb in this proximal tubule, and then we have our descending loop of Henle, our ascending loop of Henle. And you can see we're doing some reabsorption here, and the longer the loop, you know, the more you can do. And then as you move up the ascending and go to the distal tubule, now this is relationship to our Bowman's capsule, or our glomerulus. So we have proximal loop of Henle and distal. We also have both reabsorption and secretion. And then once you're here at the end of this tube, you've made urine, like you're done. So this is just a collecting duct. There's a little bit of reabsorption. I believe it's water, but mostly this is just collecting things. And so other nephrons are going to empty also into this collecting duct. So nephrotic syndrome, you can read about this. It's about the permeability of the membranes and ends up causing edema, which is fluid accumulation. All right, so 70% of your sodium ions are going to be reabsorbed in your proximal tubule by active transport. So we're using ATP, we're using energy, we're pumping sodium back into the blood. The sodium active transport requires energy, like I said, the ATP, and so does glucose, amino acids, all of these things, and even some water. Sodium potassium pumps, man, those are important. You should have heard about those many times in muscle and in the nervous system. Their job is to create a gradient, and this allows reabsorption by what's called secondary active transport. So you have sodium active transport that's going to give you energy to get glucose and amino acids and all these things back into the blood, and then you have secondary active transport that is facilitated by the sodium potassium pump. And so what happens is sodium diffuses down its concentration gradient, but there's a co-transporter protein, and so the sodium cannot move unless it also moves other things with it. And so that's how glucose and ions are reabsorbed. And that happens everywhere. And 99% of your sodium and water are going to be reabsorbed before you actually excrete the urine. Of course, antidiuretic hormone, anti-dia, don't go through. Hormone increases your water reabsorption. So it tells you anti-dia, which means do not pee. And so if you have more ADH, you won't pee. And so where's the water going if you're not peeing it out? Well, you're reabsorbing it back into your blood. And then aldosterone also stimulates the sodium reabsorption. So here's some averages for your sodium and water filtration, reabsorption, the percent, and the amount excreted per day. So you have about 180 liters of water. I still think that's crazy, filtered a day with your kidneys, and 99% of that's reabsorbed. So 1% of 180 is 1.8. So this is about how much water you're actually excreting per day from kidneys, right? And then your sodium, you have 630 grams of sodium put in the filtrate per day, and only 0.5% of that. So yeah, 3.2. They're rounding. I'm doing math in my head. Yep, 3.2 grams of sodium are released. So here's your picture. This is where sodium is actively being reabsorbed or transported from this nephron tube into the blood system. And also bicarbonates are going to be absorbed. All these ions, these things have charge. And as you increase your solute concentration, that's going to attract water by osmosis. And then chloride ions are also going to be actively transported. So we're, like I said, when we're reabsorbing, we're taking stuff that was put in the filtrate that we actually want to keep. So what is tubular secretion? It's the opposite of tubular reabsorption. It's moving substances from the plasma of the blood into those into those renal tubules that we were looking at. So we're going from capillaries to the tubules, going from blood to the filtrate. So substances are derived from 80% of the plasma that's not filtered. So remember, not everything goes into Bowman's capsule. So we have larger molecules and waste that we have to put out of the blood and get it out of the body. And active transport does that. It just is working in the other way. So what are we going to get rid of? Drugs, ions such as potassium, hydrogen. Remember that hydrogen ions are going to be important because as you have more hydrogen ions, you have a lower pH. So a fluid, an acid is anything that makes hydrogen ions. So getting rid of this hydrogen ions will regulate the pH of the body. And the potassium ions are secreted as the sodium ions are reabsorbed in the distal tubule. So remember, we already absorbed these potassiums and these sodiums. Well, we're going to get rid of some of the potassium here, and that's going to allow for some more water absorption, I believe. Let me see what this is trying to show us. It's just showing us how potassium ions are secreted. It's like, why are you showing me a picture of that? So hormones, aldosterone, and those cardiac peptides are going to affect the concentration of your urine. And so your kidneys can maintain your internal environment and therefore also control the concentration of your urine by reabsorbing lots of water, right? So if we are absorbing lots of water, our urine is going to become more concentrated. The distal tubule and collecting ducts are impermeable to water. That's so important. Star that. That's weird. The distal tubule and collecting ducts are impermeable to water in the presence of ADH. So there's the key. Remember, antidiuretic hormone says don't pee. So these cells cannot take water, let water pass through. So if you have a lot of if you don't have a lot of ADH, they can't reabsorb the water, and so you're going to have a very dilute urine. And if you think about alcohol binds to the receptors that ADH would normally bind to, and so you can't, it's kind of blocking it. So ADH can't bind, and so that's why you pee more. You have diluted urine due to alcohol, and the next day you have a hangover because you're dehydrated. However, if ADH is present, those segments do become permeable, and we can absorb the water by osmosis into our interstitial fluid and eventually into our blood, which is going to help regulate our blood pressure. And the urine is now concentrated because all the water was reabsorbed. And of course, you're going to pee less. It says, although the majority of nephrons are cortical, the juxtamedullary ones are important for that water reabsorption. They have very long loop of Henleys. Remember, they aren't just at the cortical, at the superficial edge of the kidney. They dip down into the medullary. So this is just showing if you have low ADH, so low don't pee, you're gonna pee. where if you have high amounts of don't pee hormone, you absorb all the water and you pee less, but it's concentrated with all the, waste and uric acid or urea, really, and any toxins that you put into your body. So if your body fluid is too concentrated, that means you need more. And so what's going to happen is you eventually need to stop peeing as much. And so you're going to release more ADH, and that's because of these osmotic pressure and receptors in your hypothalamus, like looking and what's the word? Monitoring the interstitial fluid in the water in the blood. And so if you have too much water or not enough water in the blood, concentrated water blood is low, we're going to release a lot of ADH, which says, hey, stop peeing because you don't have a lot of water in your blood. And eventually that water will be conserved and increase as you drink. If your body fluids are too dilute, the concentration of the water in the blood is too high, and that can be an issue. So what do you need to do? Well, you need to not release as much ADH. And so your posterior pituitary gland is reduced or blocked, and less ADH will reach the kidneys, less ADH will reach those distal tubules, and the collecting ducts don't conduct water anymore. And so they can't be reabsorbed, so you have more pee. It's not as concentrated anymore, and you lose your water. So probably the hardest part for me, in urine formation is this countercurrent multiplier. And so we see it in the nephron loops of these juxtamedullary nephrons. Remember, they're dipping down into the medullary interstitial fluid, and what they're going to do is make that fluid around the tubule hypertonic. And why do you want the fluid around there to be hypertonic? So water will, by osmosis, move out of that and make concentrated urine. So the ascending and the descending limbs, those loops of the loop of Henle or the nephron loops, they are very close together, and they work with one another. So the ascending limb is much thicker and it's impermeable to water, but it reabsorbs sodium and chloride ions. So it's going to bring these solutes into the interstitial fluid, and that makes it hypertonic, because hyper means more, tonic is concentration. So the interstitial fluid becomes more concentrated with ions. And the tubular fluid, which lost these ions but still has water, is now hypotonic. And so what happens is water is always going to leave from where there's more water to less. So it's going to go by osmosis. It says in response, water in the descending leaves. Okay, but the sodium and chloride ions don't leave because the descending limb's not permeable to them. And so what happens is you have this hypertonic tubular fluid in the descending limb. And this is the countercurrent multiplier, because every time you do it, the salt concentration increases in the ascending limb. So the vasa recta, that's a word you want to know, also has a countercurrent mechanism, but it's called the countercurrent exchanger because they don't use active transport, they're just using passive transport. So let's try to look at this countercurrent multiplier. Notice this ascending limb is thicker and it's permeable to chloride and sodium. And so what's happening here is sodium and chloride are constantly actively pumped out of the filtrate into this interstitial area. And so that makes it very hypertonic. And because it's so hypertonic, water on this side, the descending limb, it removes itself out of the filtrate into the interstitial fluid by osmosis. And so if you think about it, as water is removed and more water is removed and more water is removed, you have a very hypertonic solution in here. And then as you move through this ascending limb, the solutes that made this hypertonic are also removed. And so now you have a hypotonic fluid up here. And every time you do this, it becomes more salty in the interstitial area, which allows more absorption of this water. And then the countercurrent mechanism of the vasa recta. Vasa recta is the capillary loops or capillary bed around this loop of Henle. And so you can see this is all just passive transport. The sodium chloride actually moves into the blood over here because if you remember, it's hypertonic, and then actually leaves over here because it's hypotonic. So here are the parts of a nephron and the collecting ducts and what they do. This, I'll say it 100 times, these tables really help organize the information. So urea, I mentioned earlier, and I mentioned uric acid also. Urea is a byproduct of amino acid catabolism. So when you eat protein and you digest the protein and break it down into amino acids, you're further metabolizing the amino acids and you end up with this waste called urea. The plasma concentration reflects the amount of protein in your diet, depending on how much urea is in the plasma. So urea does enter the renal tubules through the glomerular filtration. It's small enough to go, but it's also going to undergo both reabsorption and tubular secretion. It says 80% of it is reabsorbed and the rest is going to be in your urine. Of course, the more protein you eat, the more grams you'll see in the urine. Uric acid, similar, but it's a byproduct of the metabolism of nucleic acids. And so nucleic acids like DNA and RNA are in almost everything, you should be in almost everything you eat. And so that also is going to enter the renal tubules through the glomerular filtration, and it actively transports reabsorption. 10% of the uric acid enters the urine through tubular secretion and is excreted. Now, excess uric acid, if you have too much, can create gout, where it precipitates in the blood. So you usually see this in the feet, and it forms crystals in the joints and is very painful. So what is urine made out of? Well, right, it depends on what you're drinking and what you're eating. So it can vary due to, of course, what you're ingesting, but also your physical activity. How much sweat are you, getting rid of water? But it is mainly water, and then it has these metabolic wastes, the urea, the uric acid, and even some creatine. And it might have small amounts of amino acids and electrolytes. You make about 2.5 liters per day, and so that's 60 milliliters of urine an hour. And again, those volumes can vary depending on your environment and what you're ingesting. And so here's concentrations of these different ions and substances in 3 different places. So in the plasma of the blood, in the glomerular filtrate, and then in the urine. And so you can see that all of these are leaving through the filtrate, so that's why these are equal. And then you can see what's like reabsorbed and then actually what's also secreted. You shouldn't have any glucose in your urine, right? Because you absorbed it all, reabsorbed it all. But look at the urea. You're secreting most of that. You're not getting that just from the filtrate. So a renal clearance is an actual name of a rate that they can calculate to tell how long it takes for a chemical to be removed from the blood plasma by your kidney. So it's kind of to see how well your kidneys are working and see if there's any damage or disease. And so they usually use inulin, which is a complex carbohydrate that we cannot metabolize, and also creatine. So these substances are both filtered by the glomerulus, so you'll see it in the filtrate, but neither are reabsorbed nor secreted. So that's how we can use them as a constant to see how fast the glomerular filtrate rate is. And that's called a renal clearance test or rate. So urine forms throughout the nephron and the collecting ducts like we've been talking about. And then it goes through the renal papilla to the minor, to the major, to the renal pelvis, and then eventually the ureter. And the ureter is going to move the urine to the bladder for storage, and then finally it will leave through the urethra. So let's talk about the ureters. These are tubular organs, 25 centimeters long, and they take the urine from the kidney to the urinary bladder. And so you're going to have two of these, right? One coming from the left, one coming from the right kidney to the bladder. And each ureter begins at the renal pelvis in the kidney. And so once you're at renal pelvis, you're calling it a ureter. And it empties kind of in the posterior portion in the back of the urinary bladder. And we'll see it in a second, but it makes up two of the parts of the trigon. The wall of the ureter has three layers: mucosa, muscularis, and adventitia. So a mucus coat, a muscular coat, and a fibrous coat. And it uses peristalsis to push the fluids down into the bladder. If you have an obstruction, you might have stronger peristaltic waves trying to move that obstruction into the bladder. And then you have the ureterorenal reflex. And this is where your renal arteries get constricted, and this is going to decrease your urine production in your affected kidney and cause some, more problems. Here's a cross section of the ureter where you can see the mucus, the muscular, and then the fibrous coat, and it's all surrounded by adipose tissue. If you have chronic kidney disease, it might be chronic glomerular nephritis, inflammation again of the glomerular, or the glomerular capillaries. And so you might have to have a kidney transplant or a hemodialysis. And one of my students, actually, a few of my students have had kidney stones, and they always think it's made of uric acid, but it can be a number of different chemicals that it's composed of. So they do form in the collecting ducts or in that renal pelvis of the kidney, and this is why you might see blood in your urine. Now, 60% they say of kidney stones will pass on their own, but like I said, a few of my students have had to miss class for a lithotripsy. I don't think any of them has had it surgically removed, but it is inherited, especially if you're talking about the calcium stones. But of course they say diet can also affect kidney stones. And if you're having too much calcium, that can affect those with the genetic disease. But excess vitamin D, anytime you have a UTI or blockage, that can cause some kidney stones. So we finally get to the urinary bladder, which is this distendable, the sac, muscular organ that can hold the urine. And it's going to send the urine, it gets the urine from the ureters and sends the urine to the urethra. It does contract a little bit. It's found, I saw that contacts, but it does contract. It's made of some muscle, but so they're telling you where the location is. Sorry, the anterior walls of the uterus and vagina in the female, and then posteriorly against the rectum in the males. And so, if you've ever been pregnant, you understand that it is touching the wall of the uterus, because as that baby gets bigger and pushes, you're pushing on your bladder. So here's that triangular trigoni, or trigony, I've heard somebody say, in your bladder. And so I mentioned that the two ureters are going to come in the posterior side, and then you have one in the urethra in the anterior side. We'll see that in just a second. Your bladder has 4 layers: an inner mucosa, a submucosa, Of course, the muscularis for the contraction, and then an outer serous coat, the serosa, right? Remember that serosa tissues always line body cavities and are on organs. The smooth muscles comprise of the detrusor muscle that helps empty the bladder, and then the internal urethral sphincter surrounds the neck of the bladder. And so you're going to see in this picture just the location of the urinary bladder in the male, and then here's the bladder in the female. There's that uterus we were talking about. Of course, you can't see the ureters here, but the urethra that will eventually get rid of the urine. So here's that these three holes. This is what they're talking about. So here's where the two ureters came in, and then here's where the urethra is going to go out. So that's why they named it that. And this is in a male, it looks like, right? Because there's a prostate gland and seminal vesicles. And here's a picture of it through the microscope of the inside of the bladder. You can see it's empty. That's why it's got all these folds. This is transitional epithelial tissue that makes up the mucosa lining. And then you have the submucosa lining, and then all of the muscle. And then on the outside would be the serous membranes. And last we get to the urethra, which is another tubular organ that's going to move the urine from the bladder to the outside of the body. It has a mucosa and muscularis. It has a few sphincters. Or a couple sphincters. There's the internal urethral sphincter, and it is smooth muscle, which all smooth muscle is involuntary. And so as the bladder gets filled up and applies pressure, this smooth muscle allows fluid to leave the urethra. But there is also an external urethral sphincter, and this is voluntary. It is actually skeletal muscle, and this is what you learn to control so that you don't pee on yourself. Even though this internal urethral sphincter is open, you have control over this one. And it's just mentioning that submucosa has mucus glands called urethral glands. So there are some differences in the male and female, specifically where they're located or where they're leaving and how long they are. So girls only have 4 centimeters of urethra, while a male's urethra is almost 20 centimeters long, almost 5 times longer. The female urethra will exit anterior to the vaginal opening, where the male is going to exit through the penis. And so it's actually the part of the system that is not just for urination, but also reproduction, because this is where semen will be released. And the male urethra actually has 3 different parts. There's the prostatic urethra, based on where it's located, the membranous urethra, and then the spongy urethra, which is the last part of the spongy penis. And so here you can see the spongy urethra here in the spongy part of the penis. And there's the prostate urethra. And then membranous, because it's going through these sphincters and the urogenital diaphragm. And then there's the female. Micturition is the fancy term for urination. Get rid of urine from the urinary bladder, and it does leave by micturition reflex because of these muscles in the bladder, and then they relax. That when the bladder gets filled and the muscles start to contract this reflex, that's going to cause those internal sphincters to open, and it requires you to choose to relax your external sphincter for you to actually do micturition. So it can hold up to 600 milliliters, but you're going to feel like you have to pee around 150 milliliters. And of course, it's controlled by neurons found in your spinal cord. There are some issues with micturition. We've got incontinence, where you're having trouble controlling it, and then nocturnal enuresis, which is bedwetting. Here's the major parts of micturition. So your urinary bladder distends, it fills with urine, there's stretch receptors in the wall, they're stimulated, nerve impulses travel to the muscle, which contracts rhythmically, and the need to urinate is like, oh, I have to pee. You understand. The voluntary contraction of your external urethral sphincter and inhibition of micturition reflex by impulses of the brainstem prevent urination. So you're stopping things from happening. But once you decide to urinate, the external urethral sphincter is relaxed, and then micturition reflex occurs. The detrusor muscle contracts, and all the sphincters are relaxed, and urine goes through the urethra. The micturition reflex subsides. Because you're emptying your bladder, the muscles start to relax, and your internal sphincter goes back to being contracted until your bladder begins to fill with urine again. And there's some developmental abnormalities. I was reading through these. These are really, really interesting. Like the first two, for example, are crazy. Cross-fused ectopia. You have both kidneys, so they're just on one side. And then a horseshoe kidney, where the kidneys on their inferior part of the kidney are fused together, so you have one huge horseshoe-shaped kidney. Agenesis, gen means to create, so a means without, so you did not create a kidney. Dis means, bad. Plasia is development, so this is bad development of a kidney. Hypo is low development, so it's a smaller kidney. Bad creation, an abnormal kidney or abnormal tubules. So oligomeganephronia. So meganephronia, large, and oligo. I've heard that before. I can't remember why it's reduced number, but I just thought those were kind of cool. If you were in my class, we would do a urinalysis if you wanted to, where you can measure your urine, measure the pH and compare it to a color chart, and then we look at the amount of water in your urine as well. And we said that people who have diabetes will have glucose in their urine. That's actually one way that they can test for diabetes. And there's a few things that can change your urine color and even the viscosity of your urine. That's kind of crazy. I'm trying to think of the crazy one I remember from undergrad that had to do with pee. But this is pretty funny. Beeturia. It's when you have pink pee because you ate too many beets. I think that's funny. And then not most people, but a lot of people have the smelly pee after asparagus. And both of those are genetic conditions. So you actually pass that down. I need to go eat some beets and see if I have this allele for beeturia. And of course, as you age, your kidneys start to do less, and you lose control of your bladder, and you pee on yourself, and that's giving you something to look forward to. All right, so look in your Connect and work on the assignments there for this end of the unit, and there will be a unit test for you to take.